Ciao mondo, hello world, hola mundo, hello monde, konnichiwa, ni hao, namaste. We're here today with Professor Vincenzo Cantaluppi at the Vicenza course on AKI and CRRT, and we are discussing about a very interesting topic, which is the so-called vesicles in acute and chronic patients. Can you tell us what these vesicles are and what is the meaning of these vesicles?
Yeah, sure. Extracellular vesicles are basically some microparticles that are released by activated cells, and so they are involved in cell-to-cell communication. So extracellular vesicles are formed by two main families: the exosomes that are smaller, produced by multivesicular bodies, and the true vesicles, shedding vesicles, that are larger in size. And basically they can transfer proteins or genetic materials such as messenger RNA or microRNA from a source to a target cells. So their biological activity are related to this transfer of
Signals.
of signals. So a direct activation that can be good or bad in a different setting of physiology.
So in the acute kidney injury and sepsis, for example, what is the meaning?
The possible meaning is the first is biomarker, because obviously we can check for protein or mRNA, microRNA circulating or in the urine, so potentially good biomarkers of disease state, and also some mediators of disease. We know that all inflammatory processes, including AKI obviously, are related to an increase of the concentration of plasma extracellular vesicles, and we are studying this together, so we know that these vesicles may be involved in the mechanism of microvascular injury and also tubular epithelial cell injury.
So can this make sense to try to remove this vesicle with extracorporeal therapies?
Absolutely, and what we know is that classical RRT based on diffusion or convection do not lead to a reduction of the plasma concentration of the vesicles. So basically one potential application in terms of extracorporeal blood purification should be adsorption of these vesicles.
Oh, okay. And can this also be applied to a chronic patient?
Yeah, this is another good point because it's a sort of grading on inflammation. Obviously, in sepsis we have high grade of inflammation, but we know the low grade of chronic inflammation also in our chronic hemodialysis patients. And we checked the plasma vesicles in the chronic setting, and we have about a 4 or 5-fold increase in respect to the healthy population, and they should be involved in cardiovascular injury and also in neurological alteration of CKD patients. So,
and you're planning a special study to see if absorption can normalize this?
Yeah, together with some other protein-bound uremic toxins that are involved in this cardiovascular and neurological alteration, vesicles can cross-talk with this uremic toxin. We can consider the vesicles such as uremic toxins.
Fantastic. So with this, thank you very much for joining today Cappuccinio from Vicenza, Italy, and Professor Vincenzo Cantaluppi, and have a wonderful day. Excellent.